Greetings, biochemies! Welcome to the Biochemistry Lab, where we embark on a fun, enjoyable, and fascinating journey. For this episode, we are going to perform DNA extraction using everyday materials. First, we need to wear our lab gown to protect our skin and prevent major injuries. After that, prepare the materials and apparatus needed for the experiment. To start, the test tube, stirrer, beaker, and other tools are carefully placed and sanitized. We will now prepare the fruit, dishwashing liquid, salt, filter paper, 70% isopropyl alcohol, and ice cubes. And then we proceed to the experiment. Today we are using a simple method to extract DNA from kiwi, since it also yields more DNA and provide a better glimpse into the genetic code. As we begin, we remove the skin of the kiwi fruit because it is not needed, as it's mostly dead and doesn't contain a lot of DNA. After peeling the skin, we crush the kiwi in the beaker to break some of its cells and provide a larger surface area where the DNA will be extracted. The next step is to create our extraction buffer. In a different beaker, we combine 5 grams of dishwashing liquid, 2 grams of salt, and 100 ml of water, stirring it gently to avoid bubbling. The dishwashing liquid acts like a detergent, breaking open cells and nuclear membranes, allowing the stored DNA to be released. The dissolved salt contains positively charged sodium ions that attract the negatively charged DNA, neutralizing its charge. This helps the DNA strands stick together, forming the clumps we will observe later in the experiment. While all of this is happening, we prepare 70% isopropyl alcohol in a test tube and into an ice bath for it to be ice cold as required for later use. Now we will add the mashed up kiwi to the extraction buffer and mash it again. Remember, our fruit sample should be mashed until no lumps are evident so that we can extract more DNA. But again, we must mash and mix gently to avoid making many bubbles. Next, we will heat the water bath to a temperature of 60 degrees Celsius. It is okay if it goes up as long as it does not reach 80 degrees or higher. Then we will incubate the fruit extract mixture in the heated water bath for 15 minutes, making sure to maintain the needed temperature. Letting the kiwi and buffer mixture sit during incubation helps in further breaking down the cells and begin breaking apart some of the proteins in the cells. After 15 minutes, we remove the beaker from the incubator and filter our fruit mixture through a coffee filter. We place this coffee filter paper on top of another beaker and carefully pour in the mixture to avoid spillage. This is to filter out and remove all the unwanted lumps and bits of kiwi fruit, leaving us with a clear liquid. As you can see, a clear filtrate was collected inside the beaker containing the kiwi fruit DNA. Here, we will now use the alcohol that we prepared earlier. The alcohol needs to be cold for the DNA-containing precipitate to appear faster. We will slowly pour it in small drops inside the container of our fruit mixture. At the interface between the alcohol and the fruit mixture, you should see a white cloud-like substance forming. For the post-laboratory questions, question number 1 was answered throughout the video. To answer the second question of how we are able to see the DNA, after giving the mixture time to settle, you can see 2 distinct layers, the bottom being green and clear, while the upper being white and cloudy, indicating that it's full of DNA. Question number 3. Do you think that the sample you have prepared is pure DNA? Yes, we do, but because of the time limit, we have yet to see the strands forming from the mixture to prove it. Nevertheless, our experiment ended with accurate and expected results like the formation of a cloud-like substance. The experiment serves as a hands-on exploration of DNA extraction. Through this experiment, we have successfully extracted DNA from kiwi fruit. The DNA extraction process involves breaking down the cell membranes and proteins, releasing the DNA from the nucleus of the cells. Moreover, students like us can gain insights into the structure and nature of DNA. The simplicity of the procedure also encourages further exploration and experimentation, fostering a deeper understanding of genetics and biochemistry.